Hello, everyone. My name is Junjik Song. Thank you for having me for this opportunity to talk about my research, and thank you for joining my presentation today. Today, I'd like to talk about the research entitled Oral Delivery of IL-22 mRNA-loaded lipid nanoparticles targeting IL-22 protein to the injured intestinal mucosa may be a novel therapeutic solution to treat the ulcerative colitis. Before I go into the detail, I'd like to briefly go over some background information regarding the topic of the study. Inflammatory bowel disease, which is umbrella term for Crohn's disease and ulcerative colitis, is relapsing and chronic inflammatory disorder in the GI tract that affects more than 3 million people in the US, both open develop in all ages, but mostly diagnosed under age of 35 and affects both men and women equally. As we can see in this picture, ulcerative colitis is limited to the colon, whilst Crohn's disease can occur anywhere from mouth to anus. Ulcerative colitis only affects innermost lining of the colon, while Crohn's disease can occur all layers of bowel walls. As I mentioned above, IBD affects more than 3 million people in the US and more than 5 million people worldwide. Incidence rate of IBD is known to be the highest in North America and Western Europe. Traditionally, IBD is regarded as the westernized disease, but epidemiology of IBD is changing throughout the world at the turn of 21st century, and recent data have shown that the incidence is rising and spreading rapidly in South America, Eastern Europe, and Asia as well. Recent study also shows the rapid growth in pediatric onset of IBD around the world, and the data is emerging from regions like East Asia, Africa, and South America, where it has not been reported previously. These data tells us that the IBD is becoming more serious health concern than any other time in the past. Although the etiology of IBD is still unclear, we know that from many studies that causation is likely multifactorial. Affected individuals may have chronic inflammation of the intestinal tract with imbalance of intestinal microbiota, dysregulated immune responses, or disordered gut barrier. Several environmental factors like infections and antibiotics can affect the health, and also genetic factors would increase the implication of the disease. Unfortunately, there's no cure for IBD yet. There has been development of different IBD therapeutics and has been approved by FDA and marketed for the clinical use. Depending on the symptoms and severity, clinicians move upward on this pyramid for the stronger treatment to prevent the bowel damage. Biologics therapies, including anti-TNF-alpha monoclonal antibody drug, which blocks the TNF-mediated activation of a pro-inflammatory pathway and inhibits the classical hallmark of the chronic inflammation, is widely used for the IBD patients. But it has shown the adverse effects like infections, heart failure, and could lead to the malignancies in worst cases. Next, I'd like to briefly talk a little bit about the IL-22 and its clinical relevance in IBD. IL-22 is a member of IL-10 cytokine family, and it's extensively studied relatively recently. IL-22 is a critical regulator of the epithelial homeostasis and has been implicated in multiple aspects of epithelial barrier function, including regulation of the epithelial cell growth, permeability, production of mucus, and antimicrobial proteins and complement productions. Here, we developed oral delivery of anti-inflammatory agents that targets the inflamed colon, although the micro-injection of therapeutic drugs is somewhat effective. Its clinical application has been hampered by general requirement for surgery and specific injection skills. In contrast, oral delivery offers more accessible treatment strategy that is conducive for the patient's compliance. From this aspect, development of an oral delivery method to treat the ulcerative colitis appears to have better translational value than the micro-injection method, particularly in the GI tract diseases. So next, I'm going to talk a little bit about the why the mRNA is used as a therapeutic agent in this study. I'd like to talk about what makes the mRNA a good therapeutic agent for the IBD. The advantage of mRNA therapy is the gene therapy utilizing the mRNA therapeutics that is alternative to DNA-based gene therapy, and it lies in the fact that the mRNA's effect are temporary. In contrast, DNA effect is permanent, and it also lacks any requirement for the nuclear localization of transcription, and it has no risk of genomic integrations. Lipid nanoparticles have been widely used in the pharmaceutical industry for decades because they are easier to manufacture, less immunogenic, and carry large payloads that makes them successful and efficient carrier for the various kinds of therapeutic agents, including small molecules, proteins, and nucleic acids. Recently, lipid nanoparticles were propelled into global spotlight due to the 2 approved COVID-19 mRNA vaccines. As we can see in this circular graph, lipid nanoparticle plays a vital role in many different diseases, and the tumor takes almost half the circle, and followed by the anti-inflammatory, antiviral, antibacterial, and so on. And this trend shows that the use of lipid nanoparticles in therapeutics growing rapidly, and with benefits like improved bioavailability, reduced toxicity, and the controlled drug release. These are the types of representative nanoparticles that are commonly used in the therapeutic studies. And there have been extensive studies on different kinds of nanoparticles for safe and efficient drug delivery system. But lipid-based nanoparticles offer many advantages over others, such as formulation simplicity, self-assembly, biocompatibility, and bioavailability, and most importantly, low or no toxicity in the system. For this reason, lipid-based nanoparticles are the most common class of FDA-approved nanomedicines. In our study, we used the lipid nanoparticle source from natural ingredient ginger, which is well known as anti-inflammatory food. The reason that we use the natural lipid nanoparticle is because it carries many advantages over synthetic materials. Based on our previous studies, plant-derived lipid nanoparticles show the higher specificity to the target colon through the GI tract. There was no abnormal immune responses detected, and it showed low or no toxicity. And it's flexible enough to encapsulate the various kinds of therapeutic agents. And it's also stable enough to safely deliver therapeutic agent to targeted region in the GI tract. Previously, we proved that the ginger-derived nanoparticles, so-called the GDMPs, are the excellent drug delivery system through the several publications by showing successful delivery of siRNA to treat the ulcerative colitis. And the method for making the GDMPs are well established and organized in detail in separate publications. Based on the GDMPs, we discovered that 3 major lipid components comprises more than 90% of the original GDMPs. I apologize for this uncropped data I'm showing right now, but this is lipidomics data that shows that the 3 lipids, including DGDG, MGDG, and PA, comprises more than 90% of the GDMPs. So we hypothesized that the mixing these 3 major lipids are at the ratio found in the subgroup of GDMPs would enable us to form a new nanoparticle that would retain the nanostructure of the GDMPs and may be used as a well-defined gene delivery system. So what goes inside the new lipid nanoparticle is IL-22 mRNA. The new technique used for the synthesizing mRNA is called the in vitro transcription. This picture shows the brief description of how mRNA are synthesized through the IVT method, in vitro transcription method. To elaborate briefly, first plasmid DNA was DNA with the insert. Target insert is constructed into the vector. and then it's used as the template to produce the mRNAs. And through this cloning process, newly generated mRNAs are attached with the 5' cap and the poly A tail to stabilize the structure of the mRNA, and then DNAs was treated to remove any unused DNA templates, and also phosphatase was treated to remove any 5' triphosphatase in the final stage, and then the mRNA is purified by the centrifugation to be used for the following experiment. Now we have both IL-22 mRNA and lipid nanoparticles ready to be used in the experiment, but before we apply that to this delivery system, characterization of the lipid nanoparticle is an essential part of the study, because we need to know what goes inside the system, and it could affect the efficiency and efficacy of the drug we deliver to the system. First, we measure the size and the zeta potential of the blank and the encapsulated nanoparticles. Size of both blank and encapsulated lipid nanoparticles were approximately 200 nanometer diameter, and the zeta potential was about -18 plus minus 0.5 millivolts. So the zeta potential is a measure of net electric charge on the surface of the nanoparticle, and it is crucial for determining the stability of charged particles. So next, the thin layer chromatography was used to demonstrate the lipid profiles from the original GDMPs and new lipid nanoparticles. As we can see, here the three different lipids, VGDG, MGDG, and PA, were spotted in different locations as a reference, and the new lipid nanoparticles and the GDMPs have same lipid profiles when it's compared right next to each other. And on the right side, it's an image of a scanning electron microscopy to show the physical morphology of the lipid nanoparticles. And the bottom image is the gel electrophoresis to test the stability of IL-22 mRNA encapsulated with lipid nanoparticles. So to test this, we incubated naked IL-22 mRNA and encapsulated IL-22 mRNA in RNAs for up to 4 hours, and then ran the electrophoresis to observe whether IL-22 mRNA survived or not. And this very first lane is mRNA without the RNA's exposure. And the neighboring samples are exposed. As we can see in this image, none of the IL-22 is detected when our mRNA is not protected. But we see white residues on the lipid nanoparticle stop on the top of the gel, and no destroyed mRNAs are seen on the bottom. And these two graphs on the right side shows that the size and zeta potential was stable and stable enough after long hours of incubation at the room temperature. Another important factor to check is the biodistribution and biostability. We exposed the free IL-22 mRNA and IL-22 lipid nanoparticle to harsh conditions similar to GI tract. Samples were incubated with the simulated gastric fluid at pH 3 and the intestinal fluid at pH 6.5, then ran the electrophoresis to see how it's affected. Very first left sample is free IL-22 mRNA without the exposure to acidic fluids, and next 4 samples were incubated in the fluids for the 4 hours. As we can see here, the free IL-22 mRNAs were all gone, and the encapsulated IL-22 mRNAs were mostly protected, but some of them were destroyed by the acidic conditions. On the right side, the organ distribution, is important because we don't want the lipid nanoparticles go into the unwanted regions in the system, which will increase the higher chance of abnormal immune responses and toxicity. To prove that, we gavaged IL-22 lipid nanoparticle to mice for 7 days straight and acquired the image of distribution by measuring the fluorescence. This image shows that the lipid nanoparticle only detected in the digestive tract, like stomach, cecum, and the colon. To make sure the secretion of IL-22 in circulating system, we checked IL-22 concentration in the serum as well, but there was no difference between the groups was measured, and all these characterization of this drug delivery system proves that IL-22 mRNA is well protected and the lipid nanoparticle is stable and safe enough to test in vitro and vivo models. Before we actually go into the move into the in vivo models, I want to show you that there was more reasons why our study uses simplified form of lipid nanoparticles and mRNA over DNA. The first graph shows that the transfection efficiency is almost same as newly generated lipid nanoparticles compared to the GDMPs. And since GDMP is sourced from the real ginger root, it takes more time and effort to generate the lipid nanoparticles. And there is a batch-to-batch variation. So that was our concern. So using the refined natural lipids would highly increase highly simplify the steps and eliminate any risk of instability. Next is a transfection efficiency between DNA and mRNA of IL-22. And as I mentioned above, mRNA transfection mRNA therapeutics does not require nuclear localization or transcription method. As seen here, IL-22 plasmid DNA and mRNA were transfected into intestinal epithelial cells and measured the concentration of it after 72 hours. The result tells us that the mRNA transfection is far more efficient compared to the plasmid DNA delivery. So we also did the in vivo delivery test to compare the plasmid DNA and mRNA. We gavaged the plasmid DNA and lipid nanoparticles for 3 days and euthanized 4 days after. For mRNA, we gavaged also 3 days and euthanized only 1 day after. The top graph shows the IL-22 concentration after plasmid DNA delivery, and the bottom graph shows the mRNA delivery. For the DNA, IL-22 was increased in the proximal colon after 4 days, but IL-22 level was increased in the middle colon in mRNA group, which this shows that shorter time was given to the mRNA group, but it traveled further and showed the increased IL-22 level, expression level, and this tells us that the mRNA would be a better therapeutic agent to test it in the ulcerative colitis model. Now we tested on the ulcerative colitis mouse model to observe the anti-inflammatory effect of the IL-22 lipid nanoparticles. 4 groups of mice containing 5 mice each group were treated with or without the IL-22 lipid nanoparticles, and colitis was induced by 2% DSS in water for 7 days, and treatment was only orally administered for another 7 days. Mice were euthanized 1 day post-treatment. First anti-inflammatory parameter is body weight and the colon length. Compared to the blank lipid nanoparticles and PBS-treated groups, the IL-22 lipid nanoparticle group showed the restored body weight close to the healthy group, and the colon length treated by the lipid nanoparticle with the IL-22 were also longer than the other groups. Next, we measure the fecal lipocalin-2 level, showing the neutrophil infiltration. Level of lipocalin-2 was significantly increased by day 8 after 7 days of DSS treatment, but after another 7-day treatment of IL-22, lipocalin-2 levels significantly increased, whereas the PBS and blank lipid nanoparticle group remained the same or a little bit of decrease. Also, we measured the IL-22 expression level in the colonic tissues, and we can see here that it significantly increased the all part of colon. And another important parameter of anti-inflammatory cytokines, we checked the TNF alpha, IL-6, and IL-1 beta, and they're all reduced by the effect of IL-22 lipid nanoparticles. Next, it's a histological analysis. I also showed that the IL-22 lipid nanoparticle accelerated the healing process in damaged intestinal mucosa. The top left image are the H&E staining tissues. As we can see here, that IL-22 lipid nanoparticles and the healthy groups display the well-defined epithelial structures and no leukocyte infiltration in mucosa, whereas the PBS treated group showed that the severe colonic damages like inflammatory cell infiltration and the crypt distortions. Bottom left image is the proliferation staining, and it also shows the excessive proliferation in PBS group compared to the healthy and the lipid nanoparticle IL-22 lipid nanoparticle groups. And graphs on the right side shows the significant decrease in histologic score and the cell proliferation ratio in quantitative manner. To confirm any immunological responses after treatment, we collected serum samples from each group and counted blood cells levels, including white blood cell, lymphocyte, platelets, mononuclear cells, red blood cells, and neutrophil cells. And there was no significant differences between the groups, meaning that there was no abnormal immune responses observed by the treatment of the IL-22 lipid nanoparticles. And additional immunotoxicity was tested in vitro, and the purpose of this study was to evaluate the effect of lipid nanoparticles on the human CD4 and CD8 T cell proliferation after in vitro culture of PBMC. Cells were treated with the lipid nanoparticles for 4 days, and they were collected and stained with the antibodies to be evaluated with the flow cytometry to measure the percentage of CD4 and CD8 T cell population. As we can see in this graph, population of CD4 and CD8 is no different from the control group for both viable and proliferated viable experiment groups. And the bottom graph is the quantitative measure of the images above. As well known, gut microbiota is responsible for 70% of our immune system. So we checked whether IL-22 lipid nanoparticle treatment affected microbiota composition or not. We collected the fecal samples throughout the study and analyzed the composition of it at the phylum level by whole genome sequencing. As we can see in the title of this page, there was no significant alteration of microbiota composition by the treatment. To tell you the result first. The first graph shows the taxonomic profile in the bar graph, but there was no difference observed. And next, principal coordinate analysis graph shows that the composition of trend of the microbiota was similar in all groups except the healthy group on the left. The following donut graph was generated to visualize the microbiota distribution within each group and identify identified top 7 most abundant phyla. Firmicute and Bacteroidetes were dominant in healthy group. But Verrucomycota was represented in a large portion of the population in all DSS treated groups, including IL-22 treated group here on the bottom. Next is the Venn diagram was used to display the sharing of bacterial phyla between the groups. 96 phyla were shared by all groups, and only the few phylum were distinct between each groups. And on the left, top left, the heat map and the bar graph also show the relative microbial abundance, and we found that the Bacteroidetes was the most abundant in all groups, and there were more Firmicute in the healthy group. Next, we measured calculated FMB ratio. The Firmicute and bacterial ratio is widely accepted to analyze the influence on maintenance of the normal intestinal homeostasis, because significant decrease or increase of the number means that there is some kind of dysbiosis in microbiome composition. Compared to the healthy group, there is a little bit of dysbiosis seen in all groups. This tells us that the treatment does show anti-inflammatory effect against the acute colitis, but it does not affect the intestinal microbiota. To sum up, we have developed a stable synthetic IL-22 mRNA and encapsulated within the highly efficient lipid nanoparticles originated from the GDMPs. When we orally administered the IL-22 lipid nanoparticles to DSS-induced colitis mice, inflammatory parameters such as lipocolin-2, colonic MPOs, and anti-inflammatory cytokine levels were reduced, and the body weight and the colon length were recovered to the level of healthy mice. We also tested the systemic toxicity of the IL-22 lipid nanoparticles, but biodiversity and safety were proved to be used for the therapeutics. And overall, the oral delivery of IL-22 lipid nanoparticle precisely targets inflamed colon and accelerated healing phase without affecting gut microbial composition. And we assume that this novel delivery system could be a promising therapeutic strategy to treat the ulcerative colitis. And that's it for my talk. And I want to thank all my my PI and then my colleagues who supported this research project to be done in a good way. Thank you for your attention. I'll be happy to take any questions.